You will see that I posted a bunch of links into the chat. These are just further resources that I will touch on in my talk. Feel free to go in there. I also put my email address in there if you want to follow up after the event. So I'm very honored and happy to be invited to give this keynote talk today about building and exploring the Human Reference Atlas with virtual reality. There is a vision for the usage of virtual and augmented and mixed reality for medicine. What you see here is very clearly a stock image that depicts what this future could look like. We see two medical personnel, probably surgeons or nurses in an operating room, looking at a 3D model of what appears to be a brain, presumably of the patient they're about to operate. But what becomes clear very fast is that this is obviously not real yet. This is a marketing kind of campaign imagination of what this could look like. So first and foremost, none of them is wearing a headset. Most augmented and virtual reality is headset-based or wall-based, where you need to wear some kind of spectacles to actually see it. This seems more like a hologram. Unfortunately, we don't have that reality yet. Further, if you look closer at what they're actually looking at, what kind of information can they extract from it? There is no real augmentation happening. It's just a transparent model of a brain floating in space. So this illustrates, though, where we want to go. So this is the vision for the use of VR and AR. But then you look at what we actually have right now, and it's a little more sobering. So many AR and VR applications exist for learning anatomy and physiology. That's one of the primary uses for VR for medicine currently. You can see some applications here. This is one where you have the human body in layers. And this, for example, is the musculoskeletal layer, and you can take that apart, which you cannot really do with an actual cadaver. This one seems a little more photorealistic, where you can probe the body, take certain organs out. You can also see some muscles here and vasculature also. The models vary generally in detail from photorealistic. Well, this is not quite photorealistic, but it's closer to photorealism than this one. This one is a little more on the illustrative and low level of detail side. But importantly, none of these existing applications display any kind of patient data. These are typically illustrative organs that were either modeled based on one particular patient or just based on existing textbook illustrations. So that brings me to the concept of information-rich virtual environments. This is a concept that was coined in 2003 by Doug Bauman and his co-authors. And that refers to a realistic virtual environment that is enhanced with the addition of related abstract information. So three-dimensional models which naturally exist in 3D space, but then an added two-dimensional layer that provides additional information about these 3D objects. So here on the left side, this is again a graphic from 2003. So this is, rather simple. What we see here is a human throat. We see the tongue here in the foreground. And then in the background, here and here, we see the tonsils, the palatine tonsils. And then as we go here into this other screenshot, we see a two-dimensional overlay that gives us the surface of the right palatine tonsil, which is 295 square centimeters. So this is a three-dimensional object with vertices and edges that is enhanced with a two-dimensional overlay, just giving its area size. On the right side, we have a similar concept where we have geometric primitives out in 3D space, and they in turn also are enhanced with two-dimensional information. In this case, it is a name, and then here we have some more information. It could really be anything, any kind of two-dimensional related information. So we're going to come back to this concept of information-rich virtual environments later. With this introduction about three-dimensional objects out of the way, I want to now talk about what the Human Reference Atlas is. It's, after all, in the title of this talk. So the Human Reference Atlas has been funded so far by 5+ consortia. The atlas defines the 3D space and shape of anatomical structures. These are elements, meshes that make up individual organs of the human body. The cell types of biomedical relevance that can be found inside of these anatomical structures, as well as the biomarkers that characterize the cell types that can be found inside of the anatomical structures. So it's a tree, essentially. It's a tree structure at the bottom of it all. And these anatomical structures, cell types, and biomarkers are validated and then represented and crosswalked to existing ontologies, such as Uberon or FMA, the cell ontology, and HGNC. The Human Reference Atlas also defines how new data sets can be mapped into the HRA. So the HRA consists of a series of atlas-level data sets of high quality, and then new data sets can be mapped onto the HRA. And this can, for example, be done spatially using the visible human or vasculature common coordinate framework. We're going to talk in a second about what that is exactly, or via so-called ASCT plus B tables. We're going to talk about those in a second also, but these are anatomical structures, cell types, CT, and biomarker tables, or via gene and protein expression data, which we're going to cover later in this talk also. Importantly, the Human Reference Atlas provides linked open data. So this data is openly accessible and every IRI in it resolves. And the data types are spatial, for example, the position and scale of an organ or of a cell, biological structure, such as cell types within an anatomical structure, so a distribution, and specimen or clinical, that is donor metadata such as age or sex, to preserve the donor diversity in the current HRA. Now, the HRA can be described as a series of adjectives, but these are the 5 most important ones. So it is authoritative in that there exists expert agreement and it was validated by data. We have engaged hundreds of experts across different organs to compile this current HRA 2.0. It is also computable in that it supports API queries and user interfaces, which you will see later when we demo the HRA organ gallery in virtual reality. It is also, again, published as LOD, linked open data, and it is connected to existing gene disease and other ontologies. It is also open in that anyone can use HRA data and code. There's no paywall. This is all open source. And it is also continuously evolving. We have releases twice a year as new technologies, for example, become available. The link to the HRA portal is in the chat. Now, I want to hone in on 2 major components of the Human Reference Atlas version 2.0. We have 65 anatomically correct 3D reference objects for 29 organs, so 29 unique organs, but 65 actual 3D implementations. That's male, female, left and right for some of the organs. Also 33 anatomical structure, cell types, and biomarker or ASCT plus B tables, which we'll talk about in one of the following slides. And then soon also cell type populations in 3D, for which I will give you a preview today. Importantly, there are also HRA applications, concretely these 4 that I want to cover in a little more detail today. The registration user interface, the exploration user interface, which both are 3D, the ASCT plus B reporter, which is a tree visualization, and also the HRA organ gallery in virtual reality, which you see here on the right, and which I'm going to demo later. Well, demo to the extent that that's possible here via Zoom. Here are some publications. Again, all of these are also in the chat. So let's talk about the first type of data in the HRA, that is spatial data. And I want to talk about the meat of the matter here. So to build the HRA, we need to register tissue blocks from diverse individual donors into reference organs. So organs that represent a huge anatomical variety and diversity across donors. So ultimately, then cell type populations from these tissue blocks can contribute to building cell type populations for entire anatomical structures. What you see here on the right is an artist's rendition of this is the renal pyramid and the renal papillas in the left kidney. And you can see how these are being filled with colorful dots, which represent individual cell types. So this is the long-term vision of the HRA at the cellular level. And of course, there's many hundreds of millions of cells inside just one kidney. So this is, of course, showing this at a lower scale. So the question then is, how do we register these tissue blocks into these reference organs? And it turns out that documenting tissue extraction sites is a non-trivial matter. On the left side, you can see a bisected kidney. And having a reference photo like this is the best case scenario. That's actually quite rare for us to have something like this. But normally, you would put these onto these reference boards. You can see these cutting boards in the background that have these geometric markings on them. And then you would make a mark here for the piece that you're actually analyzing, which you can see here. So you can see this is a piece of the outer kidney with a bit of adipose tissue here at the bottom. And so we use the visible human male-female data sets to model 65 3D data sets. You can see here the visible human male left kidney. Then you would see how this tissue block here would be represented inside of this particular kidney. So this all has then been productized in the shape of the registration user interface, which is, a standalone web deployed application open for anyone to use. So what you see here is the kidney, specifically the renal pyramids, and then the user can drag this representation of a tissue block into the organ. And I developed the prototype for this as part of my PhD, which then allows for users to register so far 750 tissue blocks into the 65 individual organs. Again, shown here is the kidney. And then we can go to the sister interface to the RUI, which is the EUI, exploration user interface, where you can then zoom into all of the organs and see where all of these tissue blocks have actually been registered into. So the kidney is kind of tucked back here in the back. Of course, the kidney is a very popular organ, so it is full of tissue blocks. You can also see here in the torso, all of these vital organs. You can see how many tissue blocks have been registered and how there are parts of the body that have not been mapped at all yet. So that's the exploration and the registration user interface component. I want to now talk about a term that I mentioned earlier, which is vital to the HRA being at multiple scales, and that's the Common Coordinate Framework, or CCF. So a common coordinate framework for a human body provides a unique address for each cell in the human body. Think of this as a latitude-longitude system that you can use to navigate a world map. Now, two important properties of the CCF is that it has to be in 3D because our body is also represented in 3D. We live in a three-dimensional world. And it also has to be multi-scale, so it has to work at multiple scales of magnitude. So as of the HRA 2.0, we have a total of 1,218 three-dimensional anatomical structure in the male and female body. These are the two reference bodies that we have so far. So this is where you would start. This is the default CCF level. And then if you would go one level down, you would go, for example, to the kidney, which is at the 10 it's like 10 centimeters tall. So this represents your 10 millimeters, so 1 centimeter. And then you would go down one more level. You would go down to a renal corpuscle, which is, these are 100 microns. And then you go down even further, you get to a podocyte, which is a cell, which is important in filtration, which is one of the primary functions of the kidney, of course. And you could go down even further to the nanometer scale. So this here is NPHS1, which is a gene that is important for the filtration barrier, and of course also incredibly small. So the HRA has to allow us to go across scales, from the scale that we can see with our own eyes to what we can only see with instrumentation. So at the practical level, what this means for the 3D part of the CCF is that we have a series of nested reference systems that are kind of tucked into each other. So at the top, we have the Atlas reference system, which is in part funded by HubMap, which currently contains two bodies, a male body and a female body. And then if you go even further, you get each of these individual organs, again, 65 total, and each of them, again, has their own 3D reference model, which you can see here tucked into the body. And then each of these organs has a varying variety of tissue blocks from individual donors that has been registered into it. And those, again, in turn, also have their own reference organ. And then inside of the tissue block, we have a Z stack of tissue sections. And of course, these in turn also have sections or functional tissue units that are tucked inside of them. And then if we go this all the way down, we eventually appear at a three-dimensional cell, which again has its own 3D reference system. So in theory, this allows us to register and explore these tissue blocks spatially and semantically across the macro, the meso, and finally the micro scale. All right, we talked a lot about spatial, we talked a lot about 3D. Now let's talk about the biological structure. And I want to get back to a term that is a really big tongue twister, but that is really essential for the data structure of the HRA. And that's the anatomical structure, cell type plus biomarker, or ASCT plus B table. So the background about the ASCT plus B table is about structuring knowledge with regards to what it actually does. So unstructured knowledge sources make up about 80% of biomedical knowledge. So ASCT plus B tables standardize how this information is actually captured, formatted, and labeled. It also captures knowledge about organs, the anatomical structures inside of them, the cell types that are found in the anatomical structures, and also the biomarker sets that characterize these individual cell types. ASCT plus B tables then connect each of these units of knowledge to existing ontologies, such as Uberon or CL, which some of you might be familiar. They also provide nomenclature, synonyms, and descriptions and relationships between individual entities, such as cells, anatomical structures, and biomarkers. And finally, they assign a unique ID for each unit of knowledge. So that's how ACP tables help structure knowledge. Now, as you can imagine, these tables are getting rather cumbersome to work with because they get very complex. They have hundreds of rows. And so we developed a tool called the ACT+B reporter, which visualizes each of these tables as a tree. These are basically just hierarchical trees, where each row is one unique path from the root to a leaf node. So this, for example, here is a screenshot of the ACT+B reporter visualization for the heart, where in red we have the anatomical structures, in blue we have the cell types, and in green, finally, we have the biomarkers. So the vasculature of the right atrium in the heart, for example, has these cell types here, for example, pericytes with muscle cells, etc. And then these, in turn, you can see that here with these connections. These are the biomarkers that characterize the cell type commonly. So to tie these two things together that I just talked about, the spatial and biological structure data, we have these 3D reference models. Now, instead of the kidney, I'm showing the lung. And each of these organs has a series of interconnected meshes, three-dimensional meshes, and then we can annotate those with anatomical structure IDs that then link to an ontology. And with that can be crosswalked over to an AST+B table. These organs have been custom built by a medical illustrator team. We are working with a set of amazing freelancers out in the industry, and of course with input from subject matter domain experts. And those then support the RUI, EUI, and also the HRA organ gallery. You can explore all of these organs at these URLs here. I'm going to put this into the chat later. All right, so we talked a lot about how the HRA is constructed. Now let's talk about how it is being used. So we defined 7 user stories for the HRA, which are all on the HRA portal, which I put into the chat earlier. I want to note that in the next section, I'm going to demonstrate some code that's under active development, so the user interface here will be improved. So HRA user story number 1 is to improve cell type annotations. The goal here is to use our HRA API, that's an open API that anyone can use, that can improve the accuracy of annotations for both bulk and spatial data. So in this case, we input a 3-dimensional volume inside of an organ. So in this case, for example, again, you see here the male left kidney. And as a result, you would get a table with a predicted cell type population based on what's currently found in the Human Reference Atlas. You can try that out for yourself also. It's also linked on the portal. But here is a GIF demonstration. So here you can see me define a new relocation. So I go into our registration user interface tool. I select the kidney here, I believe, or the heart. Yeah, kidney. And then I take my tissue block, place it in here. And then this is what I get back. I get back a cell summary where each row is a unique cell given a unique anatomical structure. That is the prediction that I would get. And then you can use this to validate, for example, your own cell type annotation that you would run in parallel. User story number 2 is where you that's actually quite common. Oftentimes in the whole tissue analysis process, the people actually doing the analysis are like 3 degrees removed from the people that cut up the organ. And then sometimes the spatial origin is just lost. And so HRA user story number 2 is to try to recover where this piece was taken from. So the way this works here is that you input a cell type population of a tissue block, and then you get back a goodie bag with 3 items. The predicted anatomical structure of the organ, where it likely originated from, the most similar other data sets that are already mapped to the atlas, and then an overview of all of the extraction sites in the organ that have a similar cell type population. So concretely, what this looks like is you can upload your CSV. In this case, it's just a sample CSV file. And then we get a listing of all the similar data sets, again, that we have assessed to be of sufficient quality to be part of the Human Reference Atlas. Then we have a 3-dimensional representation here of all of these heart data sets that are similar to this heart cell type population that we uploaded. And then also here a listed prediction of anatomical structures inside of the heart that seem the most similar. So we talked about using the HRA. I would now like to talk about one more HRA application, which finally also involves virtual reality, and that is the HRA organ gallery in VR. So we already had a look earlier at the exploration user interface, where you can go into the human body and you can fly around and look at all of these blocks. But of course, all of this is happening on a two-dimensional screen inside of a web browser. So we were asking, what if? What if instead of sitting next to a computer and looking at it on a two-dimensional screen, you could actually stand right next to the reference organs, which of course are modeled to scale to fit real-world sizing, and look at these models kind of as a base layer for a visualization, but in a true scale. And so this is the HRA organ gallery in VR. Just a few days ago, we finally got it published and released on the MetaQuest store, of course, free of charge. I put the link to it into the chat. So if you do have a MetaQuest 2 or 3, feel free to download and use it. So this here is an annotation of all of the things that are currently present inside of the organ gallery. And again, over Zoom, it's always hard to show something in VR. So this is like the next best thing. As you go in, you see the two humans standing here in the middle. Of course, we gave them a different, kind of like this marble type texture, just for aesthetic purposes. We are in this kind of infinite environment with a grid floor on the ground. And then we have a keyboard in the front, which has really satisfyingly to press three-dimensional buttons that then allow you to load an organ. For example, in this case here, I preloaded the heart. And then it gets deployed into this little cylinder, which you can then grab with your hand and move around. And you can actually see that really nicely here on the next slide. So this is an illustration of a user being in VR, grabbing, holding onto this heart and moving it through the scene and interacting with it, prying it open, moving it around, scaling it up and down. Here is a different view of the organ gallery from the top. So you can see here, this is our work table, which is a very simplistic cube. Here we have our two humans. One more thing you will see is if the user has selected an organ here in high resolution, they also see an outline of where it actually comes from within the body. The grid floor starts at a 1 meter by 1 meter, or about 3 feet by 3 feet. And then as the user travels up and down in the scale, which is the next thing that we're going to implement, then this is going to rescale as well. This is a closer look at the keyboard. You can see here that we have these organ symbols, so the blood vasculature, the brain, the eye, etc., etc. We also offer auxiliary functionality with the buttons up top. For example, to reset the current organ, sometimes in VR, you scale something up so much, or you move it somewhere that it's out of reach, and then you can just press this reset organ button, and then you have a nice resetting functionality. This allows you to switch between male and female, or between left and right, if appropriate. Of course, the heart, we only have one heart, so left and right is disabled in this case. And then you can see the currently selected organ is highlighted with our HRA red color. Another thing that we're currently working on is to use custom shaders to improve the aesthetics. Again, in the exploration user interface, the skin is blue. Here we chose this kind of marble texture for aesthetic reasons purely. And of course, there are also needs to optimize this application for the Quest 2 and 3. I don't know, can I get a quick hands up on who here has done any virtual reality development? Maybe a quick hands up for anybody who's ever used virtual reality before? All right, good. Okay, so there's been some usage. All right, so if you develop for virtual reality, we are targeting the MetaQuest line of headsets, which is a fantastic device, but it's also on the lower end of the hardware spectrum. So there are optimization needs in place, and we can show a maximum of 1 million triangles at once. We have to keep it at 60 frames per second for it to be released in the store, and we cannot do any post-processing or HDR. So these are restrictions that we have to work with. We also have a monitor that displays metadata for all of the current selections. So for example, in this case, it tells us, okay, we have 730 tissue blocks currently in the scene total. The selected organ is the heart, and we have 84 tissue blocks in there at present. One of my excellent students, Juhi, made this controller annotation, which is shown in VR. If you've ever for those of you that have used VR, you might know that it is sometimes hard to find the right button to press, especially if somebody stands right next to you and tells you to press a specific button. So this, when it is displayed in VR, is hopefully going to make it easier for users to understand which button they have to press. So we explain the basic locomotion and interaction. Here is an illustration of a user investigating the heart and having scaled up the heart by a factor of about, this is probably 2 orders of magnitude. So this is a gigantic heart that the user can then dive into. And this is something that you simply cannot do unless you are in virtual reality. So this is really something where VR shines. All right. So this is what you can currently do in the HRA organ gallery. I want to now talk about how we actually tie that in with data from the HRA API. So as I mentioned before, the HRA API is a free and openly accessible API to all of the data that are inside of the Human Reference Atlas that includes all of the 3D organs, all of the SG/SB tables, and all of the other digital objects that currently exist in there. And then there are specific endpoints for individual organs, tissue blocks, or an entire scene. And you can also use query strings to refine your search, essentially. So here on the left side, for example, you see the specification for the skin. So if you wanted to write your own 3D scene in any other application, you could just use the scene endpoint and you would get served exactly this data. So in the organ gallery, very concretely, we use the scene endpoint. So we use our own API to make to actually build the 3D scene in VR. The documentation is here. This provides you all 65 organs and all 700 plus published tissue blocks. So the way to read this here is this is the unique identifier for this reference organ. It is a representation of the skin. This is a so-called Uberon ID, which again is an ontology that defines the skin of the human body. The scene graph here is a URL, and it leads you directly to a GLB file. GLB is a file format for volumetric 3D models, similar to FBX or OBJ. I'm sure many of you are familiar with those formats. And then we have a transform matrix that you can then transform into a 3-dimensional coordinate to understand where the organ has to be placed in the 3D scene. So that is right relatively to all of the other organs. A tooltip, which is super handy for any kind of UI development. We're actually using that to put button labels onto the keyboard, and then opacity and a variety of other factors that you may or may not use in your application. So we are using Unity 3D to develop the organ gallery, which is a video game engine. So it makes connecting 3D objects with data rather straightforward. So here is a zoom in on the male left kidney, which we have talked about quite a lot in this talk. You can see all of the many tissue blocks that have been registered. So these are actual tissue registrations that have been registered by tissue providers into the kidney. And of course, each of these comes from a human, a donor with a sex and an age and other clinical metadata that are then easily available to us, as long as they are published, of course. And so we are capturing information about, for example, this particular tissue block here with a C# component. If you've ever used Unity before, you're well familiar with this. So this has an ID. You can see this entity ID here. This is, again, an IRI. So if you punch this into a browser, you get back a whole bunch of metadata about this particular tissue registration. And then here for the kidney, this is how we capture the API response. We have a representation of value, which is, again, an Uberon ID. This is the left kidney. So the value of this ID is the left kidney. And then we have a sex, a tooltip, an opacity, and all of these other things. So looping back to the beginning of my talk, I was talking about Doug Baumann and his thoughts on information-rich virtual environments. So on the left side, we see what we had in 2003. We had the right palatine tonsil here with this simplistic data overlay, and then these semantic objects on these geometric primitives. Now in 2023, with the organ gallery, we now have here, these are the actual tonsils from the HRA version 2.0. So these are the palatine tonsils. So this is the same organ. Of course, this is 20 years later, and we are now in a position where we can show this in an affordable virtual reality headset at a really high graphical quality. And then likewise, here now we also have still geometric primitives, but these are now richly annotated with metadata from individual donors and with cell type populations, and that are linking to assay types, and that are part of this whole rich HRA ecosystem that then links out to all of these existing ontologies out there. So it's a true piece of linked open data. I want to finish out this talk by talking about the next steps for the organ gallery and for the HRA. We recently in October held a hackathon at the Bioinformatics and Computational Biosciences branch, or BCBB, at NIAID, where we worked with a variety of virtual reality and data science experts on improving the organ gallery and really nailing down how we can make the user experience better. It was an excellent experience as a junior investigator. We also achieved pretty impressive collective performance and usability improvements. So importantly, we raised the mean frames per second. We almost doubled it, which is what we needed to get it published on the MetaQuest store. We reduced the number of triangles down from an unwatchable 4.2 million to 930K. Remember, 1 million is the upper limit that Meta gives you for the Quest 2. We improved usability of the organ selection keyboard, and we improved the lighting in the scene as well. And on top of that, we just had a ton of fun. Kristen Brown here from NIAID was the one who modeled the kidney, which I was showing pretty prominently here in this talk. Since March 2023, I also conducted a series of user needs analyses with 20+ experts. You can see these are HubMap PIs for the most part. Gloria here is a lung PI. David is a knee PI. And these are all excellent people, and I had the privilege of interviewing them for their thoughts about the use of virtual reality for exploring data sets related to these organs. We identified 3 major use cases: quality assurance and quality control for existing registrations, onboarding people to the HRA in an immersive environment, and also telling embodied data stories, which is something that is very fun to do with virtual reality. Of course, further refinement with more expert input is always needed. So the next goals then are to identify and implement pilot projects that we can take this further. We're thinking about showing microscopic data at one of the lower levels. Remember earlier when we were looking at the common coordinate framework and the scaling, and also to build, of course, always a strong network of collaborators that are interested in the research and application of virtual reality. So again, it was really fun holding this hackathon, and I just love always including this GIF here. This is us finalizing the last deliverable for this little Jumpstart Junior Investigator grant. I just always love it when the unicorns come in here in Asana. Of course, I want to acknowledge all of the workshop co-organizers with such a fantastic event. I also want to acknowledge my team here, specifically my students that I'm working with, and of course also my PI, Kadi Berner, and also my co-junior investigator, Lu Shen, who helped us optimize the organ gallery to actually get it to run and to be published on the MetaQuest store. Here are some more acknowledgements for the Human Reference Atlas in general. Here are some paper references. And finally, I would love to build, again, a bigger network of people interested in the usage of virtual reality for science in general. I would love it if you reached out. My email address and my website are in the chat. There's also a link in there on my website to my TEDx talk about living and learning in the metaverse. And yeah, happy to engage after the talk. Thanks, everybody.